Good morning to all. Welcome to the last session of the last day. The first speaker for today is Ajazul Wani from University of Kashmir, and he'll be speaking on excavating the architecture of chromatin from nucleosomes to chromosomes. Ajaz, the bell will ring at 23 minutes, and you have 2 more minutes of buffer.
Thank you, Nishana. And I would like to thank Dimple, Ranjith, and Mithun for inviting me and for this excellent conference, interdisciplinary conference on chromatin. So we are working on a couple of aspects in the lab, and today I will be sort of discussing one of the themes of our lab. So we can continue a bit. Okay, so what we have been discussing from yesterday that chromatin has an hierarchical organization, and I think every speaker showed a different version of it. This is the version which we use. So I will just like to make 2 points here. So we expect in a hierarchical organization that one level of organization influences the another level of organization. That's expected from any hierarchical structure or other way around. So, and another thing, but it hasn't been tested, the strength of this hierarchical nature of the chromatin hasn't been thoroughly tested. So another feature of this chromatin organization is that we have the nucleosomes, which when we make these cartoons or schematics, we put them at regular sort of intervals. We position them regularly. So it's not just for the aesthetic purposes, and you know that it is biological, and the nucleosomes are positioned regularly. And then we have these domains, which are sort of the units of higher order organization and the chromosome territories. So if we look at this positioning of the nucleosomes, we see that the pattern is conserved across species, although the exact linker length varies. So this tells us that this pattern or this level of organization of chromatin at the very primary level is an important feature of the organization which has been conserved. So the questions which we ask in the lab are pretty simple, like how do cells position these nucleosomes? And why is it important for the cells to position these nucleosomes regularly? Okay, so this is what I will be discussing our efforts to address this question. So we know that in the cells, there's an extensive molecular, these motors, they utilize the energy from the ATP hydrolysis, slide the nucleosomes along the genome and position them regularly. So this is an energy dependent process, and there are several ATP dependent remodelers which do this job, and one of the major players is the imitation switch or I-switch. So these proteins, these remodelers, they have an ATPase domain in the center, and they are flanked by some N and C terminal regulatory sort of or accessory domains. So what we know about the I-switch is that from some biochemical experiments and from cross-linking, mass spec cross-linking experiments, is that in the resting state, it folds its N&C terminals over its ATPase domain, and it just rests. So, but there is no structure of the entire protein of I-switch available. So this is from indirect biochemical and the cross-linking experiments. So, but in order to bind the nucleosome, this imitation switch, I-switch, it has to open up in order to bind it. So, but we don't fairly understand how this transition takes place, how I-switch opens and binds to the nucleosome. Although there is a lot of data in this state, cryo-EM as well as X-ray crystallography, in which the ATPase domain only, not the entire protein, has been the structure has been solved of the nucleosome with the ATPase domain. But we don't understand this dynamics of how I-switch transitions from this state, from a closed, what's called an auto-inhibited state in the field, to an open state. Okay, so and the next step is that once it binds, it has to slide the nucleosome after it hydrolyzes the ATP. So as I said, that we have a lot of information here, but we have almost no idea what is the conformation which I-switch attains during the actual sliding step. Because this can't be done, this is a dynamic process, it can't be done by using crystallography or by cryo-EM. So that's what I will try to take you through some of our data where we have addressed sort of this dynamics of this step and this. Okay, so for this you need an in-solution-based method to look at the conformational changes at different stages of this nucleosome sliding. So what we use is hydrogen deuterium exchange coupled to the mass spectrometry. So this is a highly simplified way of explaining it. So we have the hydrogens which are linked to the nitrogen and the oxygen atoms in a protein. And if there is a conformational change, and instead of water, if we have the D2O, so the hydrogens, okay, which are accessible to the solvent get exchanged. So we can track how many of them have got into the protein and which regions or domains of the protein they are at. So we can, bottom line is that we can track the conformational dynamics of this protein by coupling it with the mass spectrometry. As you can see that as more and more deuterium can get incorporated with time, so the mass spectra shifts towards the higher and higher mass. So we did this for the resting state of the I-switch. We took the purified I-switch and put in D2O and labeled it for different time and fragmented into the peptides, and we looked at the peptides by mass spec after different times of labeling. So we can plot this across the polypeptide. Okay, so at different times we acquired the mass spectra at different times of exchange. So you can see that from green to red there is an increase in the uptake of the deuterium. At some places it doesn't change. Okay, but what I would like you to sort of focus on is the N and C-terminal regions where we see there's an increase in the uptake of deuterium with the increase in the labeling time. So the increase in the uptake of deuterium will take place when there is an intrinsic conformational fluctuation. It opens up and it takes up more and more deuterium. So as I said, there is no structure of this entire protein available. So we use the AlphaFold predictor structure to map our hydrogen exchange data on the 3D conformation of this protein. So this is what it looks like. So at some point of labeling, like let's say after 10 seconds, you see some dark regions, light colored regions. So these dark means that there is more uptake of deuterium and light means there is less. So we can do it for n number of steps, time points at which we have collected the data. So if we cluster these conformations at different times, we see that they sort of cluster into two major groups. Okay, so one is having the less deuterium, another is having the more. So you know that uptake of more deuterium means there's an intrinsic conformational fluctuation which results in the uptake of more deuterium. So it results in the N and C terminals as well as at the interface of this protein. So what it seems or it appears from our data that I-switch is intrinsically, it's not a static structure, but it's intrinsically fluctuating between these open and closed conformations, and the relative populations will be determined by their energies. Okay, so this is one part. So once it probably opens, now it becomes competent to bind the nucleosome. So it can bind to the nucleosome. So we looked at what are the conformational changes when it binds. So again, by using the HX and MS. So here we are plotting the difference in the uptake of deuterium between the I-switch in the resting state and in the bound state. So it's the difference plot here. So anything you see out of this dashed rectangular box, so means that there's a significant change. You see many regions, there is a lower uptake of deuterium in the bound state because when the nucleosome comes, it binds to it, it shields its surfaces, it doesn't allow it to exchange, except one of the helices, which is here, which shows an increase in the exchange. So we can again map it on the 3D structure of the I-switch, and we can correlate it with the X-ray or cryo-EM structure of the ATPase domain of the I-switch with the nucleosome, and this agrees very well with what's been found. But we are more interested in what's happening at the next step, because that's what is not fairly understood, that what are the conformations attained by the I-switch during the actual sliding step. Okay, so again, we reconstituted an in vitro nucleosome sliding system, which is being very routinely used in the field, so you can detect the sliding on a native page. Okay, so we tracked the conformational dynamics of the I-switch during the actual nucleosome sliding. So again, this is a difference in the deuterium uptake between the nucleosome bound versus the nucleosome sliding state. As you can see that most of the regions are out of this rectangular box, implying that there's a conformational changes across all the domains of the I-switch during the sliding. So it automatically tells us that the conformations attained by the I-switch during nucleosome bound versus during the nucleosome sliding state, they are completely different. So this is what we found, that the conformations attained by the I-switch are completely different between the nucleosome bound versus the nucleosome sliding state. So we can map this HX data on the cryo-EM structure of only the ATPase domain. As I said, there is not a structure of an intact protein available. So we found two helices, one over here, another here, which look very important. One of them is binding to the DNA, and it's probably pulling it in during the sliding. As you can see that this color of this helix changes, it becomes more dark, implying that it's changing its conformation during the sliding. And there's another helix which is also changing the color, which is away from the nucleosome binding surface. So to find whether these conformations which we found, whether they make sense for the sliding, so we mutated some of the residues in these two key helices, which we think are very important. So as you can see, we did then these sliding assays. So surprisingly, this mutation almost rendered it completely dead. And there's another mutation, another helix, which is sort of binding towards the DNA. So it also significantly decreases the activity of the enzyme, while another mutation, although had a conserved residue again, so when we mutated it, it didn't have any much effect on the nucleosome sliding. So but to sum it up, so what we think is happening is that I-switch sort of intrinsically, it switches between these two closed and open conformations, and this conformation may be more competent in binding the nucleosome, and this binding of the nucleosome can shift the equilibrium towards this state, and then if you have the ATP and it slides. And what we found that the conformations attained by the I-switch are completely distinct in the sliding state versus the nucleosome bound state. Okay, so this is what was our insights into the mechanistic aspects of how these proteins, how these motors, they slide the nucleosome along the genome. Now, what we are also interested in, why is it important that cells have developed such an extensive machinery, okay, to position these nucleosomes, and they spend a lot of energy in spacing them. So if we change the spacing, so obviously we are interested in the 3D organization of the genome and what is the effect on the higher order. Okay, so as we are expecting, because as has been deliberated from past couple of days, this is in hierarchical organization, and we expect if we change a lower level of organization, we should expect a change in the higher levels of organization. So we use the budding yeast here model, so in which these remodeling factors were knocked out, and we did the aminase seq at 5 different concentrations, and plotted the data around the TSS. So you can see here in the wild type cells, we have a regular phasing of the nucleosomes, which suggests this is the pattern. But in the knockout strain, we can see there is a completely perturbed distribution of the nucleosomes across the genome. So given that we have used the 5 different concentrations of the aminase, so we can get another parameter out, which we call as amino accessibility score. So what you have, you plot the reads at each amino concentration, theoretically speaking, for every nucleosome, and you get a slope, and the slope of this we call as the amino accessibility score. So this was done, obviously, for both the strains, and we found there are many sites which were having the differential accessibility, and if we correlate with the occupancy of the change in the occupancy of the nucleosomes, there's an inverse relationship. So the bottom line is that the perturbation of these ATP-dependent chromatin remodelers, it changes the positioning of the nucleosomes across the genome, and it also changes their accessibility. So the point is, now we have the change here, does it have consequences, or does it get reflected on the higher levels of organization or not? So we did micro-CXL in the yeast, and this is what we have seen from Yatish's talk and from a couple other talks, that we get this pattern of these dotted pattern of the contact maps, which suggests there is this rubble conformation. So again, this was done for both knockout as well as the wild type strain to find the differences, whether there are differences. So we can have a differential contact map between the two cell types. So, okay, so we also did the high C for both wild type and knockout. So the bottom triangle shows differential contact map for the high C, and this shows the differential contact map from micro-CXL. So if we look at change in the contacts between the wild type and the knockout strain along the length of these different chromosomes, so we see there's a decrease in the short range contacts in the knockout strain. This is wild type by knockout. Okay, so there's a decrease in the short range contacts in the knockout, and an increase in the contacts in the long range contacts. So overall, this tells us that there's a change at the level of the chromosome level of organization. Okay, so if we zoom in at few places across the genome, we see that the chromatin domains, they sort of become diffused, okay, across the genome. So we quantified the insulation scores and so on across the genome. This is not just one side. So we can quantify it. We can see how much change is there in the boundaries as well as the insulation scores across the genome. So suggesting that there is some level of change in the organization at the level of these chromatin domains. So if we look at the boundaries of these domains, so what we found that they are nucleosome depleted regions. So if you can see that they are, if we compare them, the boundary is here in the knockout versus the wild type cell. So you see they are very prominent here, but they are diffused here. Now, if we look at the nucleosome status of the nucleosomes at these two boundaries, so what we see that the width of this nucleosome depleted region is slightly less here as compared to here. Okay, so we can quantify it for all the boundaries across the genome. So we classified these boundaries based on their strength. Okay, so from weak to strong. So you look at the occupancy of the nucleosomes, although the depth is same, but what it seems that there is a moderate scaling of the width of the nucleosome distribution with the strength of the boundary. So the stronger boundaries have wider nucleosome depleted regions as compared to weaker ones. And what we found that it excellently scales with the accessibility MAC score, which we determined again from the MNA-seq data. So we also looked with the ATAC-seq, and it looks similar. So the accessibility at the boundaries, so we found that the boundaries are in the nucleosome depleted regions, and the strength of the boundary, it scales with the width of the NDR, and it also scales with the MNA's accessibility. So now we wanted to investigate this correlation more precisely to see whether how the positioning of the nucleosomes correlates or changes with the change in the organization of these contacts at a much better resolution. So you know that best sites, as I showed in my slide, that this TSS is the best site to compare the positioning of the nucleosomes. So we compare the positioning of nucleosomes at the TSS of the genes whose expression doesn't change between the wild type and the knockout strain, to exclude the possibility that the changes which we see are because of the change in the gene expression. So we did RNA-seq and excluded around 370 genes who showed the differential expression between the two cell lines here. Okay, so we can plot the micro-C data contact map at a very high resolution here, the nucleosome level resolution around the TSS. So we see that there's a nice contact pattern in the wild type cells, but we see a very diffused pattern in case of knockout cells. So suggesting that the probability of contacting these different nucleosomes, this is just 10 nucleosomes, around about 10 nucleosomes from here to here. So this is a very high resolution we are looking at. So suggesting that there's a more probability of contacts when we have this regular positioning of nucleosomes, but when the positioning is perturbed, so the nucleosomes, they do not come closer to each other, or they do not contact the way they contact in the wild type cells. So, and one of the possibilities we thought to explain it is that in the wild type cells, we have a regular positioning, lying that there will be some bending positions, but if we change this, the number of bending positions are going to change. So telling us that the persistence length of the chromatin fiber here will be more as compared to the persistence length here. So now if you want to make a contact in the polymers, which are with different persistence length, the contact probability or the looping will be different. Okay, so this is what's put in the schematic here, that you have the regular positioning of the nucleosomes, we have some persistence length, and this loop end-to-end contact, or which we can say can be an assay for contact probability or looping. So it also depends upon the length, which we call as the contour length, the capital L here. Okay, so it's like if we have a necklace in which the beads are regularly spaced, and if we try to fold it, it will relatively fold easily, but if we push the beads at some place in the necklace closer and then try to bend it, it will be a little difficult to bend it. So you can be sort of explain both of these parameters and how they vary with end-to-end distance by a Kratky-Prod model. So to find this experimentally, whether the nucleosome positioning leads to the stiffness, and which in turn affects the end-to-end contacts, so we devised a sort of a biochemical experiment in which we purified the chromatin from both wild type cells and the knockout cells. Okay, so we know the knockout cells have an altered positioning of the nucleosomes, and then we removed the non-histone proteins, we digested it with a restriction enzyme, and we diluted it and then ligated it to favor intramolecular ligation. So if the chromatin piece of the chromatin is flexible, so it will come together and it will stick when we add the ligase, but if it is stiff, so it will not come together and it will remain sort of open. So we can remove these histones now, their job is done. So we add exonucleases to remove whatever is not in the circle. Okay, so because it has not ligated, because probably it was stiff, so that's why it didn't came together. So if we see more ligated DNA, so we can see there's a more end-to-end contact probability, or there's a more probability of looping. So this is the data for that. This is chromatin purification, and this is the final, what I would like to focus on this, so which is the DNA which we get in the two purifications after exonuclease treatment. We did it many, many times, and we see about twofold difference in the amount of the secular DNA which we get from the two cells, suggesting that the probability of end-to-end contact is more in case of the wild type chromatin as compared to the knockout strain. So this is what we think probably, this is one of the explanations. There can be possibly other explanations of this as well. So if you have a flexible chromatin, so the end-to-end contacts, probability of forming these contacts is more, but if we have a stiffer chromatin, so the probability of forming the contacts is less. So what I showed is that cells have this ATP-dependent remodelers. They position these nucleosomes regularly, and if and we have the higher levels of organization, but if we perturb the level, nucleosome level of organization, we see the change at other levels of organization, suggesting that there is a sort of a bottom-up biomechanical causation of the chromatin organization, which probably is expected from an hierarchical organization of any structure. So we also looked some time back in collaboration with Ranjith. We showed that if we perturb the higher order organization, we can have an effect on the lower levels of an organization. As, for example, you can imagine in the house of the pyramid of these cards, the lower levels can affect the higher levels, and the higher levels in turn can affect the lower levels of organization. So in summary, this is what we think, or it seems from our data that this hierarchical, we have tested the strength of this hierarchical nature of the chromatin organization and the ATP-dependent remodeler. So basically, this looks like shape the chromatin not only at the level of nucleosomes, but at higher order 3D organization as well. So the work was done by these 3 grad students. So all 3 of them have graduated. So I would like to thank my collaborators and obviously the funding agencies for funding, and thank you.
Thank you, Ajaz. Off to questions.
Very nice, very intricate work. It's quite interesting. So I have one question and a small comment later. The question is, there's been some work showing that bendability of DNA itself has impacts on nucleosome positioning happens. So was it completely sequence independent, or did you see some, or did you test sequence dependency?
So here, obviously, what you said in the first part is right. So, but we didn't test any sequence dependency. So we did an MNA-seq of the purified chromatin to see whether we sort of have the distinctions in the chromatin positioning after we purify it. But we didn't look into the
So the MNA-seq would also have sequence level data, right?
Yes, it has some.
But you don't see any
bias for cutting usually at the 80 regions.
So okay. So in vitro experiments, perhaps. So the other question is, I was wondering if anybody has done, or if this makes sense, if you perturb this one single nucleosome using a Cas9, dCas9 kind of a system where you can focus your mutant, one of those mutants or the triple mutants to a particular nucleosome, and then see how that perturbs that might be, maybe something to consider, unless there's something very off about that experiment.
So you mean just changing the positioning of one?
Yeah, changing the mutant in the
positioning of just one nucleosome. I don't know. It may be technically challenging.
So one can do using the CRISPR dCas9 systems. One can place one sequence. I don't know, something to think about.
Ajaz, here. Yes. So very nice talk. So when you say this nucleosome, so is there any position specific, whatever the changes you see in terms of the nucleosome positioning?
No. As I said, we didn't look with respect to the sequence whether it happens more at some sequences versus.
No, I'm not on the sequence. Let's say +1, +2 in terms of the nucleosome positioning.
Okay, you mean in terms of around TSS?
Yeah,
yes.
Exactly.
I didn't went into those details, but the +1 nucleosome behaves very differently from the +2 nucleosome in the wild type cells.
Yeah.
The max score is plus for the +1. It is negative for the +2. Okay. So but if we go compare it with the knockout strain, so the max score, we don't have a distinction of +1 and +2 there, but the max score is just positive. But the +1 nucleosome, yes, it has very distinct properties as compared to the +2, even in the normal cells.
So just one more suggestion. Maybe since you have a very nice mutant, maybe you can just transform a mini chromosome into the same mutant, just isolate and maybe just check it in the EM or something. Probably that will give you more picture in terms of how the chromatin behaves itself.
Yes, that will be very interesting.
Yeah, thanks.
Yes, very nice work. I'll go ahead. Yeah. So one question I have is, have you quantified the linker length distribution? For example, all people talk about all this 10n plus 5.
Yes. No, we haven't quantified. Obviously, the linker length distribution will be different in the knockout versus wild type cells. But in the wild type cells, I think it has been quantified to some extent in the David Miranda's paper. I think he has sort of looked the nucleosome linker lengths across the genome in the service AI. But similar thing can be done for the knockout.
Because the persistent length part it can crucially depend on the linker length distribution.
Yes,
then
it will depend upon definitely.
Just a question. So, do you see an impact of cohesin loading when you have your triple mutants? Because, I mean, since you change potentially the nucleosome occupancy, and do you see any change in or even on the high C, do you see change in the loops?
So, in the yeast, I'm not sure whether there are loops. Okay, except there is one paper, eLife 1, which they have found the loops in the G2M. But here we didn't found any loops, and the same is true with the Rando's micro-C papers. He has not found any loops there, but we didn't look at the cohesion. But yes, we looked at the H, this acetone H1 homologue in the yeast, because if its binding will change in the knockout, then that can have an indirect effect. So we found, although the concentration of the yeast H1 is pretty low in the yeast cells as compared to the mammals, so we didn't found any difference in the fraction of the H1 which was bound to the chromatin. So that could have an indirect effect, which could give us sort of the similar results, because that will change again the persistence length.
And in your in vitro, I mean, the very last, when you reconstitute chromatin, can you reach the same nucleosome density than the in vivo one?
So we didn't compare the in vitro with the in vivo. Okay, so it's not 100% same, because we have to take the chromatin through the salt wash to remove the non-histone proteins. So, but at least we can see the differences in the positioning between, I didn't show that data, so in the positioning of the nucleosomes, even after we purified it in vitro.
One more question and we'll leave the rest for the
very quick question. So in your first part of the talk, the structural fluctuation, have you thought of using Fred pairs to look at dynamic changes in the structure?
Yes, you can use the Fred pairs. Definitely you need to mutate the protein to put the fluorophores. So it will again tell you one part or about the two parts of a protein. But with this, we get sort of a global picture. People have done Fred. They have put the fluorophores on the DNA and then looked at single molecule Fred, Chauvet's lab, and how it slides, what is the pulling rate, and so on.
Thank you.
Thank you, Ajaz. We move on to the next speaker for the day.